You are welcome to the second session of cytology class. So in this session we are going to discuss on eukaryotic chromosomes, their structure and function. Eukaryotic chromosome structure refers to the levels of packaging from the raw DNA molecule to the chromosomal structures seen during metaphase in mitosis and meiosis. Chromosomes contain long strands of DNA containing genetic information. Compared to the prokaryotic chromosomes, eukaryotic chromosomes are much larger in size and are linear chromosomes. Eukaryotic chromosomes are also stored in the nucleus of the cell. Small parts or sections of the chromosomes are the genes, which code for the RNA and protein molecules required by the organisms. In organisms like human beings, chromosomes are linear, but other organisms like bacteria, chromosomes are typically circular. In prokaryotes, circular chromosome is contained in the cytoplasm in an area called nucleoid. In contrast, in eukaryotes, all of the cell's chromosomes are stored inside a structure called nucleus. Each eukaryotic chromosome is composed of DNA coiled and condensed around nuclear proteins called histones. The term chromosome was first coined by Waldeyer, and the word chromatin was introduced by Walter Fleming. In 1900, many scientists concluded the same idea of heredity that Gregor Mendel had previously. Scientists also discovered the plant and animal cells have a central compartment called nucleus. They soon realized the chromosomes were found inside the nucleus and contained different information for many different characters. Now history. It was Hofmeister in 1848 who discovered chromosomes in the dividing pollen mother cells of Tadiscantia. Strasburger in 1875 discovered the chromosomes during cell division, and Fleming described splitting of the dark stained rod-like structures during the cell division, which later he called as chromatin. Benden and Boveri in 1887 discovered that the number of chromosomes in a given species remains constant. Waldeyer in 1888 coined the term chromosome. Sutton and Boveri in 1902 proposed the chromosome theory of inheritance. In 1932, Boveri described the chromosomes as the bearer of hereditary traits. And it was Kornberg in 1974 who proposed the concept of chromosome ultrastructure by nucleosome model. Eukaryotic chromosome structure. Chromosomes are thread-like, deeply stained, compact DNA protein complex that carry genetic information in a linear sequence of genes. They are the physical basis of heredity, and they are the hereditary vehicles as they store, replicate, transcribe, and transmit the genetic information. Though the scope of the term include the bacterial nucleoid organelle genomes, viral genomes, and eukaryotic nuclear genomes, but only the last one is considered as a typical chromosome. Each cell has a pair of each kind of chromosome known as a homologous chromosome. The chromosomes are made up of chromatin, which contains a single molecule of DNA and associated with proteins. Each chromosome contains hundreds and thousands of genes that can precisely code for several proteins in a cell. Structure of a chromosome can be best seen during the mitosis of a cell division. The diagrammatic representation of chromosomes is known as ideogram, whereas the morphological representation of the chromosome of a species is known as the karyotype. So in this diagram, the chromosome consists of one chromatid, the duplication resulting in the formation of two sister chromatids. At the end of mitotic division, we'll find the formation of two daughter chromosomes. Now with respect to the chromosome number, each species contains a fixed number of chromosomes. However, a change in number may be seen in some species and is known as polyploidy. Normally, the gametic cells contain one set of chromosomes, that is called as haploid cells. The somatic cells contain two haploid sets of chromosomes. They are called as diploid cells. Haplopappus gracilis is a member of Asteraceae which shows the minimum number, where the 2n is represented by two chromosomes. In a pteridophyte by name Ophioglossum reticulum, we can see the maximum number, where the 2n is represented by 1272 chromosomes. Main parts of the chromosomes. The first one is the chromatid. Each chromosome has two symmetrical structures called chromatids or sister chromatids, which are visible in mitotic metaphase. And each chromatid contains a single DNA molecule. At the anaphase of the mitotic cell division, sister chromatids separate and migrate to the opposite poles. Chromatin is made up of DNA, RNA, and proteins. At interphase, chromosomes are visible as thin chromatin fibers present in the nucleoplasm. During cell division, the chromatin fibers condense and chromosomes are visible with distinct features. The darkly stained condensed region of chromatin is known as heterochromatin, as you can see in this picture. It contains tightly packed DNA, which is genetically inactive, whereas the lightly stained part, that is the diffused region of chromatin, is known as euchromatin. It contains genetically active and loosely packed DNA. At prophase, the chromosomal material is visible as thin filaments and they are known as chromonemata. At interphase, bead-like structures are visible, which are an accumulation of chromatin material and is known as chromomere. Chromatin with chromomere looks like a necklace with beads. As far as chromonemal coiling is concerned, there are two types. One is called as paranymic coiling and second one the plectonymic coiling. When the chromonemal threads are easily separable from their coils, then such coils are known as paranymic coils. When the chromosomal threads remain intertwined so intimately that they cannot be separated easily, and they are known as plectonymic coils. So in this diagram, The upper one represents the paranymic coiling and the lower one as the plectonymic coiling. Now another part, centromere, also called as primary construction. It is a specialized DNA sequence of a chromosome that links a pair of sister chromatids, and it divides the chromosome into two arms. The centromere contains a disc-shaped structure called kinetochore, which has a specific DNA sequence with special proteins bound to them. During mitosis, spindle fibers attach to the centromere via kinetochore. The sister chromatids are joined by the centromere. The number and position of the centromere differs in different chromosomes. The physical role of the centromere is to act as the site of assembly of the kinetochores. Kinetochore, it is a highly complex multi-protein structure that is responsible for the actual events of chromosome segregation. It helps in binding microtubules and signaling to the cell machinery when all the chromosomes have adopted correct attachments to the spindle so that it is safe for the cell division to proceed to complete and enter for the next phase, that is the anaphase stage. So in this picture, this is a metaphase chromosome, two sister chromatids along with the centromere, then kinetochore with microtubules, secondary construction, and nucleolar organizers. Other than the centromere, chromosomes possess secondary constructions. They can be identified from the centromere only at anaphase because there is a bending only at the centromere, that is the primary construction. The secondary constructions which contains genes to form nucleoli, hence they are also known as nucleolar organizer. Then another part is the satellite. Actually, it is an elongated segment that is sometimes present on the chromosome and the secondary construction. The chromosomes with the satellite, they are commonly called as sat chromosomes. Telomere, the repetitive region at the very ends of the chromosomes, are called as telomeres. They are found in a wide range of eukaryotic species, from human beings to unicellular protists. They act as caps that protect the internal regions of the chromosomes, and they are worn down a small amount in each round of DNA replication. Unlike bacterial chromosomes, the chromosomes of eukaryotes are linear, that is rod-shaped, meaning that they have ends. These ends pose a problem for DNA replication. So the DNA is very end of the chromosome which cannot be fully copied in each round of replication, resulting in a slow and gradual shortening of the chromosome. Now, at the end, functions of chromosomes. The main function of chromosome is to carry the genetic material from one generation to another. They play an important role and act as a guiding force in growth, reproduction, repair, and regeneration process that is important for the survival. Chromosomes protect the DNA from getting tangled and damaged. Histones and non-histone proteins help in the regulation of gene expression. Spindle fibers attached to the centromere help in the movement of the chromosome during cell division. Each chromosome contains thousands of genes that are precisely code for multiple proteins present in the body.